All right, guys, we are moseying right into chapter 10. This is going to cover antimicrobial treatments, so this will have a lot to do with interactions between humans and microbes. So, how do we fight back against different kinds of infections? This particular unit, Unit 3, is going to be particularly short because it only covers two chapters, Chapter 10 and Chapter 11. Now, what I don't want you to get too comfortable with is that it's a particularly quick unit, so that there's not as much to study. There's still quite a bit. It's just going to be that next exam is going to come up very quickly. So there's still a lot to study. It's just not the same amount of chapters that we're probably used to. Still put in that time, and that will pay off. So let's go ahead, tuck in, and get started. It really should come as no surprise that it hasn't been that long since we have been able to really fight back against microbial infections at all. Roughly 100 years ago, we weren't well prepared to deal with infections, and 1 in 3 children would die before the age of 5 due to some type of infection. It was kind of a big reason that people would have so many children, because the odds were you would have a child pass away at a pretty young age. So you'd kind of hedge your bets. So in the 1940s, we start this medical revolution with modern medicine with penicillin, except a couple of things start happening where the microbes start evolving to be antibiotic resistant. So that is kind of where we're at now in this post antibiotic era, where it has been so long since we started using antibiotics. We are now in a modern era where we should be able to fight microbes, but we're actually seeing a bit of a problem where we can't. We are running out of antimicrobials that are effective against microbes. Now remember, throughout this entire chapter, as well as the next chapter, I don't want you to memorize most examples that I give you unless I state otherwise. And in this example, I do want you to know these origins of antimicrobial drugs. So for bacteria, I want you to know Streptomyces and Bacillus were those first two antibiotics that we found from these specific bacteria. And we also found antibiotics from molds like Penicillium and Cephalosporium. So we were able to synthesize different antibiotics from these particular species. Make sure you know the species and know where they came from, a bacteria or a mold, a fungi. So we know that we get antibiotics naturally from other microbes. It's a natural product. And for us, we have taken that natural product and turned it into medicine, something that we can treat people who have an infection and be able to administer it and treat that infection. Now, the problem is that some infections have to be treated in such a way that those microbial or antimicrobial drugs can cause harm if the dose is too high, or if it's a particular antibiotic or antimicrobial. It can affect our cells. So, of course, the goal of any antimicrobial drug is pretty simple, and that is to treat the infection but not to hurt the patient. However, it's kind of difficult to do this because it's not always so cut and dry, so clean cut. technology-wise, we're not exactly there to have the perfect way to administer drugs effectively so that we can really get that antimicrobial where it needs to be so that it doesn't harm the patient and it only harms the microbe. Because, of course, it's going to interact with our cells. There's no way to stop that interaction from happening, the antimicrobial in our cells, if we are infected with a microbe. The other issue is that the antimicrobial can be particularly toxic to our body. And it depends on what antimicrobial drug it is that we're talking about, because that depends on what type of infection it is. If the infection is a fungus that we're trying to treat, that is a eukaryotic cell, which is the same type of cell as our cells. This can pose an issue because any type of drug that would affect a mold would also affect our cells. So it's very difficult to treat without also being particularly toxic to our host body, right? So, in short, the perfect drug to fight against microbes doesn't really exist. The way to administer it doesn't really exist. It's very difficult as far as this balancing act of antimicrobial drugs and treating microbes and not hurting the patient. Now, on top of potentially treating eukaryotic cells being an issue, there's also the issue of biofilms being a problem. Remember that biofilms are kind of those big sticky nets that trap cells of microbes into that sticky biofilm net and kind of creates layers and communities of these different bacteria or microbes that live within the biofilm. So not only is the biofilm difficult to penetrate with a antimicrobial, but there are different bacteria or microbes living within the biofilm. So whatever antimicrobial you're using might not even really matter because of those different microbes living within that particular biofilm might not be affected by that antimicrobial. On top of that, it's kind of an issue when it comes to different bacteria living within a biofilm will change how they express their phenotype, and their phenotype is their physical display. So if they change how they look, for example, some bacteria, when they are free living, will have a flagella, that little tail that allows them to essentially swim through their environment. If they are in a biofilm, they lose that flagella because they don't need it anymore. When it comes to certain medicine or antibiotics, it specifically attacks the flagella of the bacteria to kill it. Now, if that particular bacteria is living inside of a biofilm and it doesn't have a flagella, then that antibiotic is not going to be effective at all to that particular bacteria that no longer has a flagella. So there are a lot of ways that biofilms can be particularly dangerous when it comes to having to treat an infection. But we do have certain strategies for treating biofilms. So the first is interrupting the quorum sensing signals. We're going to talk about that on the next slide, so just put a pin in that for just a moment. It's essentially disrupting the communication, but we're going to talk a little bit more about it here in just a second. And then it's adding DNase, an enzyme that breaks down DNA, to our antibiotics to help penetrate essentially deeper into the biofilm and into the microbes that are within the biofilm. The thing that works the best, however, even though those strategies are after the infection, so post-infection, the best thing to do is pre-infection, is prevention. That is the best way to treat a biofilm, is to never let it happen. Don't let it start. So treating items like surgical materials, catheters, things like that, to prevent biofilms from forming in areas where we would expect to see a biofilm, potentially on the inner dwellings of medical devices, that that could form a biofilm. We would rather that not happen because we really don't have a great way to treat them. So looking a little closer at that first strategy of treating a biofilm, so interrupting quorum sensing signals, so the communication within a biofilm, um, what I need you to know is that within the biofilm, the bacteria are communicating with each other. Not crazy well. It might be very simple communication, not dissimilar to the simple communication that you have with your pet. You can understand when your cat or your dog is hungry, they go to their food bowl, they make some type of noise or indication that they are hungry or they need to go outside to go potty, something like that. It's very basic communication between these bacteria. But what we can do is disrupt that quorum sensing, that communication between the cells, as well as the communication that is sent outside of the biofilm or into the biofilm. Essentially, you're cutting the wires to where the bacteria can communicate at all. It's not a great way to treat a biofilm because essentially it just causes massive chaos to disrupt the bacteria, but it's something. It's a minor something that we can do to treat biofilms. But again, once that infection has set in, it's very difficult to get rid of. So the best way to treat a biofilm is to prevent it from even happening in the first place. That is the hope and the goal. In treating an infection within a patient, there are three things that we want to know about the microbe to treat the infection itself. One, of course, is the identity of the microbe causing the infection. The second is the degree of the microbe's susceptibility. This is called the sensitivity, and specifically it's to various drugs or antimicrobials. And then the third is the overall medical condition of the patient, sometimes called the stability. Now, in a perfect world, we would know this information, but it's very rarely ever that perfect where we know all three pieces of information at any given point in time. So, more often than not, you're trying to figure out that first piece of information, that identity. The second is you might know the identity of the microbe and you might have an idea of what the susceptibility is, but that microbe may have developed antibiotic resistance, and that can throw things off completely, which is kind of terrifying. And then that third one is probably the only one that you'll have a better understanding outside of the microbe's identity and susceptibility, and that is the status of your patient. It's important to understand this one. In fact, I'd put a star next to this one and tuck it back in the back of your brain to always keep in mind the stability of your patient as you are treating them. If your patient is teetering on the brink of life and death, and you treat them for the microbe, you give them that antibiotic, and it turns out that that particular microbe is able to release endotoxins. And if you remember endotoxins, we talked about them when we were talking about cell walls of our microbes. So if a microbe is able to release endotoxins, that means that the microbe has died and it releases the endotoxins as a fine. If I'm going, I'm taking you with me. So if they release those endotoxins and your patient is already on the brink, yes, you've treated that bacteria, yes, but you have potentially already now killed your patient because their stability was already on the brink. Granted, this is not always avoidable. If you have to treat that condition, and their stability is on the brink, but it is literally, you have to treat it or they will die, then, it's kind of a gamble you have to take. But if you can hold it off, let the patient get better for a few days, or at least a few hours, something. Give them some time to reach some form of stability before you treat that microbe, for the risk of the bacteria releasing those endotoxins. That might be the move. So not all bacteria are going to show antibiotic resistance. Here are a few that do. So that Staphylococcus species, that's going to be the species that ends up causing MRSA, where that is a particularly big problem in hospitals and healthcare fields. So we do see quite a few of these. That gonorrhea species, that is going to be the superclap. So we are seeing certain microbes develop really strong resistances to antibiotics and have little to no susceptibility. So there are some microbes that we do have to test for their susceptibility in order to treat them. Some we don't. Some we know that we can treat them right away. Others, like this list, we do have to look at their susceptibility to make sure that we are treating them properly. However, there are a handful of other microbes, like fungi, and protozoans that generally do not show a resistance to antimicrobials, and they can be treated pretty easily. I say that. We're going to talk about how to treat those guys here in a little bit. So this is one of two ways that we're going to talk about how to look at susceptibility in microbes. This is the Kirby-Bauer technique, or the disc diffusion method. So what we do is we make a lawn of bacteria across a petri dish, and then we drop a few discs of antibiotics around the dish, and we look for zones of inhibition, sometimes called kill zones. So those kill zones are going to be those areas of no growth, where the bacteria is not going to grow around the antibiotic. Now, it's not completely a great comparison together, but usually the bigger the kill zone, the better the antibiotic, but that's not always a good comparison or a good method to kind of go with. But what we can see, at least for that picture in the top, at the top picture there, the bottom left disc has a very big kill zone, as opposed to the disc that is directly above it, kind of around, I'd say like 10 o'clock, right? That guy doesn't have any kill zone, so that tells us that that antibiotic is not going to work. So that is the purpose of the Kirby-Bauer technique, is to look at what antibiotic is causing the death of that particular microbe. The second way to test susceptibility is going to look at both what the Kirby-Bauer technique looks for, so what antibiotic is going to kill the microbe, But it's also going to look at a second thing. What dosage is required to kill that microbe? So we use these plates that have a ton of wells, and we put a different amount of the drug, and we dilute it more and more as we go through the tubes. So as we add the microbe, we're going to see probably a color change, or something is going to happen in there where we see a difference where the microbe is being killed off or not in that particular well. So the tube or well with the smallest concentration of the drug that visibly does not allow growth is going to be the one that we want to use. So not only are we seeing which antibiotic is going to work, it's also going to show us what dosage is required to kill that particular microbe. So this is called the minimum inhibitory concentration, because that's the end goal. We want to know the minimum amount of that particular drug. We want to know how effective it can be in that minimum dosage. So this is called a MIC test. It tells us, again, two things: both what the Kirby-Bauer technique told us, which antibiotic is going to work, as well as what lowest dosage is required to kill that microbe. So you would want to choose an antibiotic that, one, kills the microbe, and two, requires the lowest dosage. You wouldn't want to have to take the highest dosage of any antibiotic, because that is going to cause all kinds of results in us, like side effects, not good ones. Now, sometimes we do everything right to treat a patient and they're still sick. Sometimes they get worse. What does that mean? What happens? Well, it can mean a handful of things. One, it might be the inability of the drug to diffuse into the body, or parts of the body that are infected with that microbe. For example, if you're having digestive problems and you're given a topical treatment, you're not going to be able to treat a digestive infection if you rub something on your skin. You'd have to ingest that particular medicine or be injected with something in order for that medicine to actually work. So it has to be administered correctly. The second is maybe there's a resistant microbe to the infection that didn't make it into the sample collected for testing. This one's kind of a by chance. We missed something in diagnosing, but it's not necessarily the fault of the diagnoser. It's kind of just by odds that we didn't catch the microbes that were causing the infection. The third is an infection caused by more than one pathogen, called a polymicrobial infection, is causing a secondary infection. So this is going to be, this one and the next one are going to be our top two contenders for when things go wrong, when a treatment fails. What's happening? Usually there's a secondary infection happening. That first infection made it to where that second infection is able to come in and start wreaking havoc. So that is typically what we're going to see. And we're going to keep coming back to that term, polymicrobial infection. So I'd maybe underline that, highlight it. We're going to keep coming back to it over and over through this unit. For that outpatient problem, that fourth one, that is going to be your biggest uphill battle. So this is going to be where people stop taking their antibiotics before the round of their antibiotics are over, because of course they start feeling better. So they're going to stop taking their antibiotics and put it up on the shelf because save it for a rainy day when you start feeling bad, which is the biggest no-no. And as a healthcare practitioner, you want to make sure that you really push that they need to finish their entire round of antibiotics. Do not save it for later. That is a big, big no-no. So if you want to put a little shorthand here, outpatient problems, it's just people being people, not understanding the importance of finishing their medicine all the way through, or taking their medicine properly. So it's essentially just people not following instructions or not understanding the instructions properly. So that is going to be one of your biggest issues in the healthcare field. In choosing the correct antimicrobial drug to affect any type of infection, there are a handful of things we need to take into account, but the biggest one is going to be whatever that ratio of the dose of the drug that is going to be toxic to us, because we have to take it, right? We are going to be introducing that drug to our cells in some capacity. There's no two ways around it, compared to the minimum effective dose that is going to cause that microbe to die, right? So what we call this ratio is the therapeutic index, or the TI. It's going to be composed of two numbers. I'm going to go to the next slide because it shows it just a little bit better. The number on the left is going to be the toxicity of the drug to us, and we want this number to be higher and also substantially different from the number on the right. The number on the right is the toxicity to the microbe, and we want that number to be really low. So, which of the following ratios is going to be the best option? The biggest goal is for these numbers to be as far apart as possible, and for the number on the left to be really high and the number on the right to be really low. So we want that third option, that 100-to-1, because the number on the left means that we need 100, I don't know, maybe milligrams or something for the drug to hurt us or kill us, versus 1 milligram is going to affect the microbe. Does that make sense? I hope. So let's talk about interactions between drugs and microbes specifically. So the goal of antimicrobial drugs is to specifically attack those microbes. How they do this is through selective toxicity, meaning that the drug is going to kill or inhibit the action or synthesis of certain molecules within a microorganism, but not of vertebrate cells. So it is going to be selectively toxic to parts of microbes, is essentially what we're saying here. Now, the issue that we run into is that not all microbes are going to have all of the specific molecules or features of a bacteria, like peptidoglycan or a specific size ribosome, because those are easy targets. Those are something that only bacteria have. However, if we look at something like protozoans or fungi, excuse me, or helminths, those are going to be types of eukaryotic cells, so that there are very few targets, molecularly speaking, for us to attack so that our drugs are effective against the microbe, but also don't hurt our cells. Viruses are also a problem because they use our cells to kind of make their own acellular structures. So they use our cells' machinery to reproduce. So they also have very few targets that we can attack. So those are kind of the problem children. Those protozoans, fungi, helminths, they're eukaryotes, and viruses, they kind of hide using our cell's machinery. So those two are the issues. Bacteria are easier to attack because they have very specific features that the drugs can focus in on. There are 5 basic mechanisms for antibiotics to attack a bacterial cell. I do want you to know all 5 of these and what they're doing. So these 5 basic mechanisms for how an antibiotic is going to attack a bacterial cell. The first is going to be the inhibition of the cell wall synthesis. So this is the most common mechanism that we use. So things like penicillin are going to do this process. So it's doing exactly what it says it's doing. It's going to prevent the cell from making the cell wall. And remember, the cell wall is a pretty important part of the cell. It's going to keep everything in, and it's going to keep the things that it doesn't want in the cell out. So if the cell can't make what is essentially the skin of the cell, then it can't keep things out, it can't keep things in, it can't function as a cell. This is a very good mechanism. The second is the inhibition of nucleic acid synthesis. This is going to attack that transcription of DNA replication. So this is, transcription or DNA replication. So this is going to prevent DNA and RNA from being able to, what will eventually be, make proteins, right? So if you don't have DNA or RNA within the cell, proteins can't be made. Those structures that the cell will need eventually will not be able to be made because DNA and RNA are essentially what makes an organism that organism. So you are getting rid of that genetic material. This is another good one, but it is harder to utilize. The third is the inhibition of protein synthesis. This is kind of going a step further from that second mechanism. So it is inhibiting gene expression. So we absolutely cannot make those proteins. So it's stopping that transcription translation process at the point of translation instead of stopping it at the DNA, RNA part, you're stopping it at the RNA protein part. The fourth is the alteration of cell membranes. This one's kind of a fun one, kind of goes hand in hand with the first one of inhibition of the cell wall synthesis, except this one doesn't prevent the entire cell wall from being made, it pops holes into the cell wall. The same problem arises though from the first mechanism, where the outside of the cell comes in and the inside of the cell goes out. So if you're popping holes in say a boat, right, it's going to start to sink. Same process here. And then the fifth is going to be the inhibition of metabolite synthesis. This is going to prevent metabolism of the cell, but the problem with this one is that it's going to affect our cells as well. And prevent our cells from being able to metabolize and synthesize different nutrients as well. So that one's kind of a sticky one because it's also going to affect our cells. But these are the 5 mechanisms of how an antibiotic is going to work against a bacteria. Moving on to some stuff that maybe you've already heard of. There are 2 major groups of antimicrobial drugs, broad spectrum and narrow spectrum, and they do exactly what you can kind of imagine from the title. The broad spectrum drugs are going to be kind of known as a shotgun method. They are effective against more than one group of bacteria, meaning they have a very wide range of bacteria or microflora that they're able to affect. An example is tetracycline. These have a little bit of a caveat to them though. So with broad spectrum drugs, they can also affect our home team bacteria, and that is not a good thing. That's pretty bad. So it sounds great, but it's going to hurt us in the long run. It can also create really strong mutants of bacteria because it's killing everything, right? It's essentially killing off wide ranges or wide groups of bacteria all at once. And what's left is the strongest of the strong bacteria, which can be terrifying and is one of the big reasons why we are currently in this post-antibiotic era, because we have used broad spectrum drugs way too much. Now, narrow spectrum drugs are very precise. They are targeted, and they target specific groups of bacteria, or even specific species. So I'm going to butcher this term, and I'm very sorry. I think it's polymyxins, but this is a very specific antibiotic that is very specific to particular bacteria. These guys are great. The problem with these is you have to know first precisely what you're treating, and if you don't, then this won't work. On the exam, I am probably going to give you a chart, much like this, that shows different groups of bacteria and which antibiotics that, range across these groups are going to affect those particular groups of bacteria. So we have our tetracyclines, and remember, tetracycline is going to be a broad spectrum, and we can see that it's spanning across gram-negative, gram-positive, chlamydia, Rickettsia. So essentially, what I would have you do is look at this particular chart, and I'd say, okay, which one is the broad spectrum drug? You would tell me tetracycline. I will not give you something that is kind of an intermediate, like streptomycin or the sulfonamide. That is kind of a mean one to give you because you aren't as proficient at knowing if that is a broad spectrum or a narrow spectrum. Meanwhile, you should be able to look at that polymyxin and know that because it is only affecting a small group, a small amount of the gram-negative bacteria, it is a narrow spectrum drug. It is precise versus the tetracycline, which is spanning across all of those different groups. It is a broad spectrum. So be able to tell the difference if I gave you a chart like this. All right, I'm sneaking what is technically a third kinda major antimicrobial group, drug group into the mix, and that is penicillins. Now, the reason why I didn't bring this up with the two major ones is because it's only somewhat kind of a third group. Penicillins are no longer what we originally had as penicillin. Originally, it was a narrow spectrum that affected gram-positive bacteria, but it was susceptible to microbial counterattacks. Now we use it more like a broad spectrum, and we have affected penicillins to the point, lab-wise at least, that there are a ton of different penicillins essentially that are used. I think it's like some 20 plus different kinds of penicillins, but they are pretty broad spectrum at this point. But it's only because we've modified them because we've had to. Microbes started becoming resistant to penicillin within a year after we started being able to use penicillin. So we had to start modifying our antimicrobials. pretty quickly because that evolution started happening within bacteria very fast. So this is kind of a third group, a little bit sort of kind of, but I would know the other two a little bit better. Know penicillin as kind of a third group. Okay, from here on, we are talking about treatment. What I need you to know is why are certain things hard to treat? That's going to be a big thing. So, in treating fungal infections, for example, ringworm, and remember you don't have to know examples, it's just sometimes nice to keep in mind an example. It can make life a little easier. Something like ringworm. The reason that fungal infections are difficult to treat is because a fungus, a mold, is a eukaryotic cell. And remember that eukaryotic cells are the same type of cells that we are. So, in treating those cells, we have to use drugs that are going to affect our cells, which can be a really big issue. I'm sure you can kind of make that connection there. So we can't use any type of antifungal agent that is going to be so strong that it's going to absolutely tear our cells apart. And for the most part, what we see is fungal infections are going to be more superficial, something like ringworm or athlete's foot, something growing on our skin or our nails. What we do is we use some type of medication that speeds up our cell replacement so that it causes the fungus to fall off along with the skin cells or the keratin that is being sloughed off. So if you ever have ringworm, if you've ever had ringworm, you put on a topical treatment to your skin, it causes the skin to get really dry because it's accelerating how your cells are growing so that that fungus essentially just sheds off of your skin, which is kind of gross to think about, but that's how we treat a fungal infection, typically. Scooting right into protozoal infections. Another issue with protozoal infections, other than they are absolutely terrifying, is that they are also eukaryotic cells. Much like those fungal infections, they are an issue because they are the same type of cell that we are. Again, same issue. If you use a drug that is too toxic, it's going to affect our cells. So we have to be very careful. However, when it comes to protozoans, the kind of biggest known protozoan infection is going to be malaria. That is a parasite that is carried by mosquitoes. It is a worldwide disease. It's one of the oldest diseases, and we've had a lot of research into making anti-malarial drugs, as well as treating mosquitoes and getting them to not be able to carry malaria. There's a lot of research that has gone into specifically this protozoal infection. So again, make sure you know why it's hard to treat protozoan infections, and it is because protozoans are the same type of cell as we are. They are eukaryotes. On to helamins. When they cause an infection, this is going to sound like deja vu, they are also eukaryotes. So that is going to be another issue, why it is hard to treat those parasitic worm infections. Another thing, and I want you to know this when it comes to helamins, is that they are going to have stages. So these worms have larval stages as well as egg stages and adult stages. And when they're in these different stages of their life within us, because they're living inside of us, they can be difficult to treat as far as what stage they're in. Adults, essentially what we do is you take some type of medicine that causes violent diarrhea. Um, this is going to clear out everything, including the worms. Typically, it's going to get the eggs out too. You may have to take something else to make sure that everything gets treated, but for the adults, that's a pretty good flushing out of the system. When it comes to the eggs, however, certain drugs might work on the adults but not the eggs, or might work on the eggs but not the adults. It just depends on what type of worm we're talking about and what stage it is that we're talking about. But for the most part, good rule of thumb, violent diarrhea is going to kind of clear out the whole system, get everybody out. You may have to take supplemental drugs afterwards just to make sure there's nobody left over. Um, but yeah, there you go. Uh, we've got helminthic infections. Again, they're eukaryotes and they have different stages. That's why they are hard to treat. On to viral infections. These guys are unique in their challenges because they use our cells for most of their metabolic functions, for reproduction. It can be difficult to treat viruses. And in fact, it's so difficult to treat viruses that we don't really have that many treatments for viruses. We kind of just treat the symptoms and you have to ride it out. The best way that we have found to treat a virus is prevention, kind of like with the biofilms. The best way to not get a virus is to give yourself a vaccine. It will prevent you from getting that virus in the first place, so we don't have to find a way to treat it. So things like polio, we try to eradicate these viruses after finding those vaccines so that they just go away. Sounds kind of like a cop-out, but really it's not. It's just finding a way to fight the virus, making it go away before it becomes more of a problem, right? Same with measles. However, measles is starting to kind of come back, which is kind of a scary thing. We're going to talk more a little bit about that kind of fear surrounding vaccines eventually, but that is essentially what is happening, is that people have more of a concern about vaccines than the virus itself, it seems, which can be kind of a scary combination of which do you fear more, the vaccine or the death by that virus. But then you have things like the seasonal flu, where we have a vaccine for that. Again, that is just to preemptively try to create this prevention of the flu virus from getting around our population. So again, it all comes back to prevention of the virus itself, because we don't have a way to treat the virus once someone is infected. Now, I just harped forever about how we don't have a way to treat a viral infection. We do, but they're not good. So these are the three major modes of action for treating a viral infection. But let me explain why they're not good modes of action for treating a viral infection. The first is blocking penetration of the virus into the host cell. However, this means that host cell can't have anything else get in either, virus or otherwise. What is that host cell? Those are our cells. So we're blocking the virus from getting in, but we're also blocking the cell from doing anything else as well, metabolically, right? It essentially creates a dam around the cell so that nothing else can get in and nothing can get out. This is not good for our cells. So in the long run, that one isn't great. The second is blocking transcription and translation of viral molecules. And what does a virus use for transcription and translation? Our cells. So essentially, you're just blocking our cell from doing gene expression. And our cells need to do gene expression. So if you're preventing the virus from doing gene expression, you're preventing our cells from doing gene expression. And then the prevention of the maturation of viral particles. So this is going to stop uninfected cells from building what they need to in order to stop the infected cells from building more viral particles. So it's kind of a double-edged sword in all three of these major modes of action to treat a viral infection. Yeah, you stop the virus, but you're hurting your own cells in the process. So there really is not a good way to treat a viral infection. Again, writing out the symptoms, that is the best way to kind of go through once an infection of a virus has happened. But the best thing we can do is prevention of a virus. So we keep bringing this up, and that is that microbes have evolved to have antimicrobial resistance. So what that means is that a microbe cannot be affected or killed by a drug that would have otherwise killed them. So they have drug resistance to antimicrobials. What we are living in now is known as the post-antibiotic era, which, not to put the absolute fear of it into you, but you should be a little bit worried, not even a little bit, you should be a lot of bit worried, that we can't treat microbial infections, which are very common, with antibiotics. So what we need to start talking about now is how did that happen? How did microbes develop this resistance? Is it our fault? And in part, the answer is a little bit yes. So resistance to penicillin, and remember, penicillin is kind of the OG antibiotic, was used in 1940. Resistance to penicillin was discovered in microbes in 1940. That's not a typo, guys. That is what happened. It took less than a year for bacteria to become resistant to the only antibiotic that we had at the time. So in the 80s and 90s, we started seeing a lot more research in these treatment failures, kind of getting amped up. So let's talk about this antimicrobial resistance a little bit more and how did it happen? So in microbes, we can see that drug resistance can come about in three ways. So I would maybe title this slide as, how do microbes become antimicrobial resistant? Well, there are three options. Number one, genetic versatility and adaptability of microbial populations. That is a fancy schmancy way to say, and I'd write this down, mutations. We are saying that it is a mutation that has occurred. So it's kind of like the heavens opened up and said, okay, microbe, I'm blessing you with the ability to become antimicrobial resistant. It is a mutation because remember, mutations are random and unpredictable. So that genetic versatility is literally kind of a blessing when it comes to bacteria or microbes in general, right? The next is intrinsic nature of a microbe. So this means that the microbe just naturally doesn't work against other things. So it's when resistance is a fixed trait of a microbe. So all microbes are intrinsically resistant to antibiotics that they themselves produce. But the thing is It's whether or not they're introduced to an antibiotic that they haven't seen before. So this one is less important. We're not going to talk about this one again. We are going to focus on the first and the third. So let's talk about that third one. That is acquisition of resistance. I want you to remember horizontal gene transfer. And if you didn't get it the first time, didn't get it the second time, let's talk about it a third time. Remember, horizontal gene transfer is going to be that acquiring of a gene from another cell, whether that is through conjugation, transduction, or, oh my gosh, and I'm blanking on the third one. Don't worry about it though. But essentially, it's that acquiring of that gene from another cell. Yes, horizontal gene transfer. So suddenly obtaining that ability from another cell, acquisition of resistance. We are going to focus just on genetic versatility, which is mutations, and acquisition though from here on out. Don't worry about that second one too much. Now, I know it seems like potentially humans in the pursuit of treating infections are the primary cause of antibiotic resistance. Now, that is not the case, and we shouldn't take on all the guilt, although we have made it substantially worse. What has happened in the natural world is that bacteria just naturally become resistant to antibiotics, because where do antibiotics come from, or antimicrobials in general? It comes from other microbes or other organisms. Antimicrobes or antimicrobials are naturally occurring substances in the world. So for example, so that we can shed just a little bit of this guilt here, in a cave in New Mexico in 2012 that was unearthed, we discovered 93 new species of bacteria that were cut off from the surface of the earth for millions of years. In this cave, we see this natural selection, this development of bacteria being resistant to antibiotics that we currently have, which is terrifying, mind you. But how did they get that way if we've never met them before? So this bacteria has never met a human. How did it become resistant to antibiotics? And it is because there is competition occurring inside of this cave. So, bacteria naturally becomes resistant to antibiotics through the exposure of antibiotics, whether that is through mutations or acquisition. This is a naturally occurring phenomenon, much like, and I have this little example here, these blind cave fish. They naturally lose their eyes because they don't need them in these caves. It is a naturally occurring phenomenon. So, whether we got involved with bacteria or not, they were going to become resistant to antibiotics, regardless of our finagling and interacting. We've just made it worse and probably faster. So as I said, we are going to focus in on that genetic versatility, which we are now going to call mutations, those spontaneous mutations, as well as the acquisition, which is essentially horizontal gene transfer. So conjugation, transformation, and transduction. Sorry, I couldn't remember it earlier. My brain just blanked. I knew it was coming up, but here it is now. So again, we have these two primary ways that microbes are able to develop resistance to antibiotics. We are going to focus in on just these two for now. So starting with our spontaneous mutations, Again, remember that when it comes to mutations, that these are going to be unpredictable and random. It is very rare upon rare upon rare to see a mutation that is one, beneficial, and two, that pops up when you need it. So, these spontaneous mutations that lead to antimicrobial resistance, please know this is a very rare occurrence, but it still happens within bacteria to develop that antimicrobial resistance. Still really rare, but it can still happen. Remember that the other way for a cell to become antimicrobial resistant is through acquisition. So this is acquiring those genes that make the cell itself resistant to an antimicrobial. We are going to call these resistance factors, or R factors, so conjugation, transformation, or transduction. This is going to be through plasmids or transposons, those jumping genes, right? So it is more likely that this is going to be that method that happens for a cell to become antimicrobial resistant, the sharing of genes, if you will. So even though cells can't communicate from one cell to the other, like, hey, Bob, do you want to become antimicrobial resistant like me because I didn't die? Oh, sure, Steve, I'd love to become antimicrobial resistant. They can't communicate like that. That is not what's happening. They're still working together to become resistant to antimicrobials. So we are going to talk a little bit more about that, but before we do, we're going to talk about something that's a little bit newer called persisters, but then we're going to kind of jump backwards into the how. So like I said, a little bit more recently discovered are these things called persisters. These are a physical response when antibiotics get into a bacteria's space. Persisters will cause the microbe to essentially go into like a hibernation or a sleep or shut down, a temporary nap. And then once the antibiotic is gone, the cells will end up waking up. And essentially, if you can imagine it, all of the antibiotics are gone, the danger is gone, and everything is perfectly fine. So this is just a phenotypic, not a genotypic response to that danger of an antimicrobial in the environment. So they just go to sleep, wake up, and all of their problems are gone, which is kind of cool. For the bacteria, not for us, but these are persisters. Another recently discovered antimicrobial resistance feature has been these interfering RNA or RNAi features, which have the ability to decide to remove or turn off the feature that makes the cell susceptible to the antimicrobial itself. So they'll turn the feature off and then turn it back on when it's safe. So it's called epimutation when they silence the gene, and an epigenetic event when the entire reversible mechanism happens. So kind of interesting. Now, we've discovered this in fungi primarily. So that antimicrobial is going to be an antifungal, not an antibiotic. So this is kind of a bad example, but I want you to think of it like if we introduce an antimicrobial, and the cell was able to turn off its ability to make a flagella, because that is a common feature that we will attack on a microbe, is the flagellum. So the reason that's a bad example is that fungi don't have a flagellum, but just bear with me as we talk about this. If the cell was able to turn off its ability, that gene expression, to make a flagellum while that antimicrobial is essentially in its area, well, that's kind of cool, because then that antimicrobial can't affect it because that is what it attacks. So it's removing the target on its own cell. So that is that interfering RNAi, which is kind of terrifying to think about, that it can just modify itself. But again, we've only seen this in fungi. And again, kind of a new thing. All right, let's stop there for now for our Chapter 10, Part 1. Join me for Chapter 10, Part 2, and we will finish up talking about drug resistance and what we're doing to try to kind of get away from this threat of being in this post-antibiotic era.